Welcome to the Deep Dive. We dig into the sources you send us to pull out the key insights. Today, we're tackling a treatment that's, well, both incredibly effective and, frankly, a bit strange on the surface. Fecal Microbiota Transplantation, FMT. It's basically using the body's own microbial world to fight off a really nasty infection, specifically recurrent Clostridioides difficile infection, or RCDI.
And this isn't some rare thing.
Hmm.
RCDI is a major public health issue. It's an aggressive superbug, attacks the gut, causes a lot of hospitalizations and deaths. Our sources point to something like 14,000 deaths every year just in the U.S. because of this.
14,000. And the cost financially, just as serious. You factor in the repeat hospital visits, the treatments that don't always work. It adds up. We're talking around $4.8 billion a year for the U.S. healthcare system. It's staggering.
Yeah, and a big driver of that cost is, well, the standard antibiotic approach often fails. Patients get stuck in this cycle. About a quarter see the infection come back after the first treatment. And if you're unlucky enough to relapse twice, your chance of it happening again jumps way up, maybe to 60%. Mm-hmm. The antibiotics just can't reset the gut microbiome properly to keep C. diff down.
Okay, so you've got this aggressive bug, treatments that often fail. It sets the stage. Our mission today is to really unpack why FMT is such a surprising breakthrough here. We need to look closely at the different ways it's delivered, because that choice impacts effectiveness, patient experience, cost. And critically, we have to grapple with the very real safety issues and the regulatory hurdles that come with using, well, human biological material as medicine. Okay, let's unpack this.
The effectiveness numbers for FMT and RCDI are, well, they're truly disruptive. For those patients who've already had 2 or more recurrences, the cure rate we see reported is around 90%.
90%. Compare that to what you just said about antibiotics.
Exactly. It blows the standard antibiotic therapies out of the water for those difficult cases, where you might only see a 30 or 40% success rate after multiple recurrences.
That's transformative, going from maybe a 60% failure rate to a 90% success rate. And the cost savings must follow pretty quickly then.
They do, because you stop the cycle of recurrence. One study looked at this and found FMT saves, conservatively, about $17,000 per patient, mostly by avoiding those really expensive repeat hospital stays that define chronic RCDI.
Makes sense. But those savings depend on actually getting the transplant material into the patient effectively. So let's talk about the delivery options. We know there are a few viable ways to do it right, which gives patients and doctors choices.
That's right. The basic idea is simple. Get a filtered stool preparation from a healthy, very carefully screened donor into the patient's gut. You can deliver it to the lower GI tract, usually via colonoscopy or sometimes an enema. Or you can go through the upper GI tract, maybe with a nasojejunal tube, a tube through the nose down past the stomach. And increasingly, we're seeing effective, validated methods using capsules, basically swallowing pills containing the prepared stool.
Right, and the capsules immediately sound less invasive. Colonoscopy, putting a tube down your nose, those involve procedures with their own small but real risks. The endoscopy itself. But do the capsules work as well? Is there a trade-off between comfort and efficacy?
That's the million-dollar question, isn't it? And luckily, we have some solid research here. A key randomized clinical trial directly compared oral capsules to colonoscopy for delivering FMT in RCDI patients. And the results were pretty striking. The capsules achieved a 96.2% success rate.
Okay, that sounds high. How did the colonoscopy group do?
Also 96.2%. Statistically, they were non-inferior, meaning the capsules worked just as well as the colonoscopy delivery.
Wow. So identical effectiveness, that really shifts the conversation towards the practical side, doesn't it? Cost, patient experience.
Absolutely. And the differences there are quite stark. In that same trial, the cost for the capsule procedure was around CAD $395. Compare that to CAD $1,120 for the colonoscopy route. Big difference.
Huge difference. And what about how patients felt about it?
That was revealing, too. When they asked patients, 66%, 2/3 of the capsule group said the experience was not at all unpleasant.
Yeah.
For the colonoscopy group, only 44% said the same. So patients clearly preferred the capsules. Less invasive, less unpleasant.
So capsules are just as effective, much cheaper, and patients prefer them. Sounds like a clear winner, but is there a catch? Does colonoscopy offer something capsules can't?
There is one critical advantage to the colonoscopy route that you lose with capsules. It's a diagnostic one. See, the symptoms of RCDI, bad diarrhea, abdominal pain, they can look a lot like other serious conditions like inflammatory bowel disease, IBD, for example.
I see where you're going.
Right. When the doctor performs a colonoscopy to deliver the FMT, They get to actually look at the colon lining. They can take biopsies if something looks off. It's a crucial safety check. They can rule out these other potential diagnoses that might need completely different treatments. With capsules, you miss that opportunity entirely.
That's a really important point. So it's a trade-off between the convenience and preference for capsules and the diagnostic safety net provided by colonoscopy. This brings us squarely to the biggest challenge with FMT overall, safety. We're transferring live, complex, biological material. There's an inherent risk, isn't there, of passing along harmful pathogens?
It's the paramount concern, absolutely. Transferring stool means potentially transferring stool-borne diseases. That's why donor screening has to be incredibly rigorous. Think about the standards used by stool banks like OpenBiome. It's intense. We're talking a 109-item clinical assessment with a doctor, plus around 30 different tests on their stool and blood.
And even with all that, what percentage of people who want to donate actually pass?
It's tiny. Only about 6% make it through the entire screening process.
6%. That just highlights how difficult it is to find suitable donors and how seriously they take the screening. It must be a huge logistical challenge for the banks.
It is. And even with that level of rigor, sadly, it's not foolproof. The FDA has had to issue warnings about this.
Yeah, I saw those safety alerts. Really serious adverse events, right? Infections linked to FMT products, even from pre-screened donors at a school bank. We're talking things like pathogenic E. coli EPEC and STEC strains.
Exactly. And Ceftec, the shikatoxin-producing E. coli, that's the one that can cause really severe food poisoning, even kidney failure. In the cases the FDA reported, 4 out of 6 patients who got the contaminated product needed to be hospitalized.
And tragically, it didn't end there. 2 patients who already had complex medical conditions died after getting FMT from a donor who was later found to have STEC. It's just a devastating reminder of the potential consequences if a pathogen slips through.
And what's really crucial to understand is how it slipped through in one of these instances. The donor linked to the STEC infections had actually tested negative before donation using a standard type of test, an immunoassay. But later, when they went back and tested retained samples using a more sensitive method, nucleic acid testing, the samples were positive for STEC.
So even the rigorous testing missed it initially. That really drives home the fundamental challenge here, doesn't it? Human stool is just incredibly complex and variable from person to person, even in the same person over time. Guaranteeing consistency like you would with a manufactured drug seems almost impossible.
It is inherently difficult. And that variability, that complexity, leads us straight into the regulatory puzzle. The FDA decided to regulate fecal microbiota as both a drug and a biological product. They're trying to fit it into the existing framework used for standard chemically defined pharmaceuticals. This raises an important question. What are the consequences of doing that?
Well, it seems like trying to fit a square peg in a round hole almost. Experts point out it creates this tension. On one side, you risk underregulating the safety aspect because this stuff is so much more complex and variable than a typical drug. It needs constant vigilance.
Right. And on the other side, you risk overregulating access. If one company manages to navigate the incredibly expensive drug approval process for an FMT product, they could get market exclusivity, monopoly protection.
Meaning they could charge potentially thousands and thousands of dollars for treatment.
Potentially, yes,
Yeah.
for something derived from essentially donated human stool.
And that creates another huge safety risk, doesn't it? If the approved product becomes unaffordable, patients who are desperate facing this horrible infection might turn to do-it-yourself FMT at home. Using stool from friends or family that hasn't been properly screened at all. You see guides online for this stuff, apparently with tens of thousands of views. It's a real danger.
A very real danger. The goal of regulation has to be safe access for patients who need it. That's why some policy experts are pushing for a different approach, sort of hybrid regulatory model, something similar to how the FDA handles human tissues, like cord blood, for example.
Okay, how would a hybrid model work? How does it balance safety and access better?
Well, the idea is for RCDI, where we already have strong proof that FMT works, you'd focus regulation on the process. Mandate extremely strict donor screening and material handling standards. Ensure safety through process controls. But critically, you wouldn't grant market exclusivity for the basic FMT product itself for RCDI. This keeps it accessible.
Okay, so safety through process standards for the proven use. What about other potential uses? People talk about FMT for IBD, metabolic diseases, even depression.
Right. For all those other currently unproven indications, the traditional rigorous pathway would still apply. Companies would need to file investigational new drug applications, conduct proper clinical trials to prove safety and efficacy, just like any new drug. So it maintains strict safety standards across the board, ensures access for the proven life-saving use in RCDI, and prevents monopolies while still demanding solid evidence for any new applications.
Wow. This has really been a deep dive. FMT is clearly a powerful life-saving tool for RCDI, but getting it to patients involves this complex balancing act, weighing the convenience, cost, and patient preference of capsules against that vital diagnostic safety net you get with colonoscopy. And all of this happens under the shadow of significant safety risks inherent in the material itself, and within a regulatory system not really designed for something biologically complex.
So what does this all mean? Well, the conversation around FMT is definitely moving forward. Many researchers, I think, see current FMT using whole stool as maybe a necessary first step. There's a lot of work happening now on developing targeted synthetic bacterial treatments. Companies like Seres Therapeutics are working on this. The ultimate aim there is to figure out which specific microbes are doing the heavy lifting and then grow just those beneficial bacteria in a lab. That way, you could potentially get the therapeutic benefit without any of the risks associated with using complex, variable, hard-to-screen donor stool. You'd have a defined manufactured product.
That really brings us to the final thought for you, the listener, to chew on. If the ultimate goal is to safely give the patient the beneficial microbial function they need, where should the main focus be? Should we keep trying to perfect the delivery and safety screening of inherently variable and risky human stool? Or is FMT as we know it now maybe just a crucial bridge technology, something vital for now, but destined to be replaced once we have targeted, manufactured microbial therapies that offer the same benefits with greater safety and consistency?